to cover part of new modalities in drug discovery because as science is moving, there are a lot of new things are coming. So earlier it used to be back old days when there was no medicine, modern medicine, pills and all those things. We used to have the herbs and all those things. Now there are pills came, antibodies came, and proteins came, and then there are a lot new things are coming every day. You heard about vaccines. There are messenger RNA vaccines, only RNA strand. So I'll try to cover as much as possible. It is going to be a little bit longer, I hope. So let's move on. So these are the new therapeutic modalities. I have to stand here because he's making a movie on that. So in last 20 years, there are 17 new modalities has been discovered because of the new medicines, new diseases has come up. So earlier it used to be a small molecule. When I started my career in India and US, it was only a small molecule. I did not touch anything else. Then there was antibody drug conjugate that came, and I'll answer some question on that. Then targeted, this is a brand new technology which is being used, which is called targeted protein degradation. So disease are caused by proteins, and if instead of inhibiting that, can you close that door? So this is a brand new, so I will, because I am working right now, other than a small molecule, I am working on antibody drug conjugate and targeted protein degradation. So I will give more information about that. Other than that, there are RNA targeted small molecule. So a small molecule RNA vaccines, everybody has used that. There are many RNA vaccines. Just last week or so, President Putin from the Russia, he announced that he has an RNA vaccine for to treat the breast cancer. So it was in news, and they are going to give free throughout the world. RNA therapeutics are there. Macrocyclic peptides are there. Macrocyclic peptides means, if you know peptide, it is sometimes open chain. So if you make a ring of that, or if you connect one end to other end, it becomes cyclic. So that we call it macrocyclic peptide. Gene therapy and editing. This is a very, very well-known modality, but it is very expensive. Gene therapy is very expensive because you are going to change your DNA, or you are going to change DNA ex vivo, means outside your body, and then inject that DNA into your body to cure the disease. And editing is CRISPR editing. We know everyone knows that CRISPR is an editing tool to edit DNA of a disease molecule. Engineered cytokines. I don't know, you remember, cytokine storm was a word used during COVID time. It means that inflammation. Cytokine is inflammatory proteins. And when people used to get sick, doctor used to say, oh, he has a cytokine storm. Means cytokine was generated by body and they were attacking your own body. So you have to contain that. So there is engineered cytokine which will not, attack your body. Cell therapy is another modality, and oncolytic virus is people are working on for 10, 20 years. So these are the couple of new modalities which I will discuss. So black color one I will discuss briefly. Red color ones I will discuss a lot, because I cannot, if every modality need one lecture like an hour. So you can go ahead and read all those things. You will get the slides. Okay, so antibody drug conjugate. So since I'm working on that, not only I am working, this has become very popular, and it is very safe medicine also. So people started working on that, and I will give you the history how it starts. Then mechanism of action of disease cell, component of ADC and its assembly and means synthesis. I will give you a flavor of how it is synthesized. Actually, I have an approved project on this. And one of my, because in Saudi Arabia, we have to have high school student who will come and work with me because they have, this is called Mohiba program. I think we should start something like that here. So high school student right from the school, summer they will come and they will work in our lab. Usually they don't work in the lab because we don't allow them to touch the chemicals, but they will see or shadow somebody. So just I got that one. So this project I gave to one of the student, and he is in the next step. So there are steps where people go to from one place, they call it Talmida, IBDA, and then ISEF, which is International Olympiad. So this person has been selected, went to next step of the program. Then approved ADC drug, then use of ADC beyond cancer. I will not talk about that because I think I will cover only cancer part and then summary. So history of antibody drug conjugate, and you can see that in 1913, Paul Ehrlich, he describes the concept of magic bullet. What is magic bullet? Magic bullet means the pill which will treat everything. So you take one pill and you are cured from all diseases. So that was the proposal. It was by Paul Ehrlich. He won Nobel Prize also in 1910. Okay. And this was concept, and you can see from there nobody has done anything here to here. Means concept, he gave a concept, who cares? Nobody wanted to do anything. Then in 1950, somebody started doing few things, and then slowly it started picking up. ADC proposal for was there on 67. And moving on, there was one person who asked about that which drug was withdrawn. So Pfizer made this drug called Mylotrag in 2000, okay? And this was a ADC with colchicine and another antibody with a linker. I will explain all those terms in next slide. So what happened that this drug was on market for 7 years. So from 2000 to 2007, it was on market. And in 2007, it was withdrawn. So he was asking that it is possible, can somebody withdraw that? Yes, this is a huge conglomerate, big company. They have to withdraw because there was some issue with dosing and it was toxic also. Why it was toxic? There was a reason for that, because the drug colchicine was releasing before it reaches the target. So the concept for antibody drug conjugate, which will come in the next slide, that how it works, means you can use colchicine like Taxol alone, you can antibody alone, and it will be cured, like doxorubicin, etoposide, mitoxantrone, all are used alone also. They are toxic, but they treat the patient. Why to do antibody drug conjugate? So the next slide will explain to you. This is the mechanism of antibody drug conjugate. So what happened there on the cancer cell, there are antigens. So if you have an antibody specific to that antigen, it will bind only to cancer cell, okay? Now, okay, so what? It binds that, right? So what we do, that we make a toxic drug which is called colchicine, for example, or doxorubicin, for example, and we have to connect that drug to a protein which is antibody. You can see there. So this is antibody drug conjugate, and you'll see in next slide how it is being built. So you can see that this is the antibody drug conjugate. It has an antibody which is very specific to the antigen. This is antigen here, okay, and the drug is attached to antibody. So it is like a targeted targeted attack, I will call it, okay? This is a missile which is going to target timely only at that part of the cell, okay? So that's why we call it warhead, okay? Warhead is a word is being used on TV a lot. So this becomes a warhead for this cancer cell, okay? So the antigen is here, this antibody with this drug, which is linked to here, binds to antigen, and then the process called internalization. So the antibody drug conjugate, okay, I'll go back. It internalized, okay? So internalized means it gets into the cell, okay? Then mechanism starts. There are lysosome, endosomes. So now this drug has to be released from the antibody. Then it will work only, right? So there is a process that the antibody will be destroyed by lysosomal enzymes. So because the antibody is a protein and there are protein enzymes there, they will destroy the antibody, then it will be released. The drug will be released inside. So what happened, I don't have to take calciciamycin and doxorubicin directly IV. We have to bind it to the antibody. Antibody will go only to cancer cell. It will attach to the antigen which is on cancer cell, internalize. Once it is internalized, once it is inside the tumor cell, as I mentioned here, it is inside the tumor cell. Now it is other cells are safe. The healthy cells, they will not bind the antibody. So these antigens, yeah, one more point I want to make sure, I think you will ask me. These antigens are only developed on the cancer cells. They are specific. So researchers, they figure out how these antigens, which are the antigens which are on the surface of the cancer cell, not on the normal cells, okay? So it is clear and mechanism. Then what happened that it is by lysosomal, degradation. This is degraded. The drug is released. You can see the drug is released here, and some of the drug they are going to, and mostly cancer drugs, mostly cancer disease is due to DNA. So it will go to DNA, bind in different mechanism, whether it is intercalation, whether it is alkylation or whatever it is done, and then this will destroy the nucleus and the cell that, the cancer cell that happens, okay, everybody. But sometime these, we call it, they usually don't, they release early, okay. So we call it bystander effect, but it happens very rarely, okay. So no modality is 100% safe. That's the message I want to give you, okay. So ADC, this is the mechanism of ADC. When you get this slide, please study every part of that and you will enjoy how it works, okay. Now coming to the little bit of construction, okay. Yesterday I said that, and in my first lecture also, I think I said that molecules are built like brick by brick, like this building is built, carbon, hydrogen, nitrogen, you have to connect. Same here also, it will, we have to do that. So this is antibody. On antibody, there are few attachment sites. There are two kind of amino acid, lysine, which has NH2, means amine, and other is cysteine, which has sulfur. So both can be used. The chemists are smart, they know how to do that. So I will explain in few slides. So the attachment site is fixed now. Antibody, we know, attachment site, we know. Now linker is very important, and this is the chemist, because if you put a strong, if you put a, like a linker like a steel rod, it will not release the your drug. So you have to have a linker which can be, which can release this drug easily. And there are mechanism of that, and I will discuss little bit more on that. And then the drug, and then you have to choose a nice drug. For every cancer, there are different drugs, so you have to choose a different drug. Okay. Um, uh, for breast cancer, people use doxorubicin. Also for colon cancer, there are other drugs. So you have to choose the different drug for that. Okay. So antibody, this is target a well-characterized antigen with high tumor expression. So I already explained that the antigen is expressed on tumor and it should be high so that the antibody can bind to that. Maintain all the properties upon conjugation to the drug. That's another. So antibody is a protein. When you do some chemistry on that, it can change. So you have to think about it, how you are going to do that. Maintain non-specific binding also. So, you know, there are amino acids. There are two amino acids usually we use, but there are other amino acids. So if you use a proper chemistry, it will bind only to cysteine and lysine. Okay, attachment sites through cysteine and lysine. I already explained that. Variable drug antibody ratio. This is an important part. It will be explained, and it shall be site selective conjugation also. So it will be site selective means it should be on this part only, which is FC part we call it. Okay. So the conjugation should not be here and there, because this is the part which is going to bind to the antigen. Okay. So that's important. Linker, this will be cleavable and non-cleavable. Those both kind of linker we use. Stable and selective release of the drug. This is very important. If you use a weak linker, as soon as you inject, it will release. You don't want that. You want a linker which will circulate, goes to the cancer cell, bind it, get inside, and then release. As simple as it is. Okay. So, and drugs, this will be highly potent. So in this case, we use very, very toxic, highly potent drug which cannot be given IV, okay, for the cancer. So as bad drug as possible you use here. That's what I will say. Okay. They should be non-immunogenic, because immunogenicity is a problem. If drug is already immunogenic, you will start getting, before even reaches to target, you see the side effects. Could be linked to and this drug should be linked to antibody and defined mechanism of action. So this should have a defined mechanism of action, means it should be doing on the nucleus, like you can see here, microtubulin, DNA alkylations. So it has to do something within the cell. Okay, so evolution of antibody, I already talked about it when it started. So the technology started in 1975, although Paul Ehrlich already told in 1910, but we started doing that. And the first approval was a very, very weak, very small doxorubicin antibody. It was not even antibody. I will not call it antibody drug conjugate. Then Marion et al., he started making some. So this was by Morrison in '86. Jones et al., he made a dadizumab, and then real antibody started in 1990 and then 1994 by these two gentlemen. Okay, so the classification of antibody is also important. When you see drug, you will see Z-mab, Zumab, S-mab. So why it is like that? So I have already mentioned, when you see on something OMAB in literature, it means it is a mouse antibody, okay? Because we start working with mouse. We don't work directly to human. So we have to make a drug for mouse first, and then make a drug for human. Okay, Zimab is like 35% mouse and 65% human. When Zumab, actually, these things I have to remember myself. I really forget sometimes that what is the meaning of Zimab, Zumab. It looks like some Chinese language, but you have to remember that. In literature, you will see a lot that if you see something, they call it Zumab, it means this is 5% mouse and 90% human. And then UMAB is 100% human. So for human, most of the time, 90% time, we like to use humanized antibody. So there are production procedures, so we use only humanized antibody. But in certain cases, mouse antibody also can be used. So it is not off the hook. Okay, so when you do that, okay, yeah, that's important. I just in previous slide I talked about immunogenicity because mouse has a different makeup of antibody with different amino acid, and human has a different, although they are same antibody. So immunogenicity it is a problem. The mouse antibody will not be very good for human. It will be immunogenic, and there will be side effect. It will kill you. But as soon as you go to human side, human antibody, that works very well. So, but you have to start for making antibody drug conjugate. You have to start with the mouse. So that's the point I want to make. That why we do that. Okay, conjugation strategies. Sometimes it is site specific. Sometimes it is random. I already talked about almost half of that. So random is like lysine is on the surface of antibody, cysteine is on the surface of antibody, so we do that. So in case of lysine, it is all over, but cysteine is only 4 of 5. So we break the bond of cysteine and then try to bind that. So then we have control. How many drug per antibody is going? We don't want 20 drug per antibody. We need only maximum 4. So this is what we call is DAR, or drug antibody ratio. Okay, so in some cases there is no cysteine, means very little cysteine on antibody. So we engineer, which is called protein engineering. So we put more cysteine in the antibody, and it will not affect its work. Then there is enzymatic modification. We have to do that sometime, and we use an enzyme called transglutamase. Then conjugation to non-natural amino acid also. So there are 20 natural amino acids. There are many non-natural amino acids. So sometime we use that also, and by protein engineering we put in the antibody just to make sure that our drug is attached to that. Sometimes, lysine and cysteine is not enough. So some other amino acids you have to use. Okay, so linker strategy, this is more chemistry part. So linker can be two kind, cleavable, non-cleavable. I said that it should cleave, but in 1 or 2 cases we have seen that if it is not cleaved, even then it works. So it is very strong binding antibody drug conjugate, and it does not need cleavage. So it just goes inside the cell and then bind to the endoplasmic reticulum or DNA or Golgi apparatus, and it works. And the whole molecule is destroyed by lysosomal enzymes. Enzymes are within this cell. Okay, there are many enzymes are there. So so we call it acid labile, which is hydrazone. Those who have little chemistry background, they can understand better. We have valine and cysteine, and that is what I am using in my research. Because I don't have big resources to do other. So I am using valine and cysteine, and we have synthesized. We call it maleimide, and this part we have already synthesized. And this cysteine formation is very easy on that. So we design that we can work in the lab properly, okay? We don't have full resources like pharma company has. Then mercaptoacetyl caproyl antibody. This is just length of carbon. That is why it is called caproyl. Maleimidocaproyl. This is another one where you form the amide bond. So it depends on your drug. You should have a handle to bind to this linker. So we have to maneuver here and there and see which can be good for other. And then MCC, and there are many more. There are many new one came. I wanted to just emphasize that I am using this one at present. Okay, I cannot use everything. Okay, so payload. Payload means the toxic drug like doxorubicin. I'm just mentioning few of them here so that you understand how diverse the anti-cancer compounds are. That's the point I'm trying to make. It is not like one compound. Okay. It is like, look at the structure of the compound. and selection criteria is very important. As a team, we sit down and we see, okay, which one is we can use for this particular antibody. I worked on in my previous life. I worked on prostate cancer. This we call it mesothelin in Bayer, and that was a different story. Here I am doing for breast cancer. So you can see there is a auristatin, doxorubin, nimorubicin, and PNU. They are very, very toxic drug. If it is given by injection IV alone, it will have a very bad side effect. So that's why we have to make very specific delivery part. You can call antibody drug conjugate as a delivery also, drug delivery, which is part of the course, okay? Because you are using a specific delivery vehicle, which is antibody, to deliver your compound to the cell, okay? So everybody get there. So this is CBI dimer, and you can see their potency, 0.0007 nanomole, means it is 7 maybe picomolar or femtomolar. It's so toxic. This much amount is more than enough to treat your cancer. So that's why I wanted to show these structures that you should know that how potency, look at this auristatin, 0.07 nanomolar means it is 0.7 picomolar. You cannot imagine what is picomolar, okay? So it is very, very toxic drugs, and they are all used in the practice in many places. The other one is tibulicin, 0.01 to 0.8, very potent molecule. Everyone has a very different structure, very different structure. And this is statins, and this is catechiamycin. I talked about catechiamycin. This is very toxic drug, and this was the part of Milotrag, which was in market and then withdrawn. And then again it is back. Last year it is back because someone found out that the dose which was given early was very high, and it was given one time. So the clinician said that, okay, let's divide 2 milligram dose into 5 times in 24 hours. Still it works. So it is that this is a one drug in the history of FDA. It was there, it was withdrawn, again came back. So this is called Milotrag. So that is for one of the. So this is the compound. You can see the complexity of the structure that was bind to the antibody, and it was done by one of our Indian fellow actually in Pfizer. I know him. It is just we have to have the resolve and the courage to do that, and he did that. Okay, I just listed, I'm not going to read that. This is the payload. This is the very latest Nature review which came. So you can see earlier it was like 1 or 2. I used doxorubicin and auristatin in my work. But now there's a lot of choices you can use that. So it is not one, because research has grown, so it is a lot of choices now. So this is the list, and I'm sure that you get the slide and you can read this literature also. And this has given the payload classes and what they do actually, whether a mode of action is given there, like inhibition of tubulin. Inhibition of tubulin polymerization induces DNA double strand break. Similarly, DNA alkylation. DNA cross-linking, DNA intercalation. So these are all words. When I started working, my postdoc was on anti-cancer compounds, so I learned that alkylation is different than intercalation, and linking is different than so DNA is a prime target for the cancer, and then stabilization of topoisomerase inhibitors. This is an enzyme which is usually expressed in the cancer patient. So that also can be targeted. So these are all very new. You can read that. Okay, assembly of components in of now how we actually make. So we have the antibody, okay, and as I told you that I like personally, so that's why I put it there. I like personally that cysteine should be the target, okay, not the lysine. So first we have 4 cysteine bonds. In antibody, usually we have 4 cysteine bonds. Why I target cysteine? Because I can control. That's all it is. Okay. So there is a partial reduction by TCAP. This is a chemical compound. Easy thing to do. Once you have open sulfur, you use enough TCAP to reduce only one bond. You need little bit more to reduce another bond. Okay. So this is again, I say control. You can control it. Then we conjugate with the maleimide here like that. This is called a Michael addition in chemistry. So we do Michael addition here, and then after Michael addition, either you start making work here. There are things that you can make the whole drug from here and just do last step, or you can start making different way. Lysine is also there, so you can do through this. So there are many ways. So I just simplified the whole mechanism. It is not as simple as look here, but this is the way it is done. So in two cases, two antibody drug conjugate which is on market. One is Adcetris, which is heterogeneous ADC, and this is Kadcyla. I think it is already available in India also. I know that some of the companies they are making already. So these are the two approved. I just wanted to show you the complexity of the structure. They are sold in like 10 to 15 billion per year in many places. In India, both of them are available. What they are, Adcetris is Brentuximab vedotin. This was approved in 2011. The antibody is Brentuximab directed to CD30 that is expressed on HL and LCL. Attachment site for me is thiomaleimide caproyl. Caproyl is the long length chain of the carbon. Spacer is PBAB, which is here, right here. This is your linker part, and this is the warhead or cytotoxic drug. Okay, so this is Adcetris. This is made through sulfur conjugation to antibody. In case of Kadcyla, antibody drug conjugate, this was approved in 2013. The MAB is for breast cancer, HER2. Attachment is again through lysine, and we have to put another part into that, and then okay. So these are the linker. We have to increase the length so that it can internalize properly and go inside the cell. And the drug is mertansine, very potent, very cytotoxic drug. Okay, so these are the two I wanted to show you the enzyme example for that. Okay. How much is okay? So I have to rush a little bit. These are the approved antibody. You can read that there are 13 antibody drug conjugate used. Now, in like 2020, the first was made from 2020 to 2000. 2000 first was made, and 2007 nothing was made other than that. That was withdrawn. So from 2007 to 2025, 13 antibody drug conjugate made already, and you can see that how new it is. Okay, everything. And in last year there were 4 of them they approved. So this is a good area if you have resources to work on that. And so this is the 2025 data which I am giving you. The second is clinical trial landscape, and there are 200 clinical trials are going on right now. So using antibody drug conjugate. So this is about antibody drug conjugate. I will move on. I told you that I will talk about protein targeted protein degradation, or PROTAC, what we call it. This is very important. This can be done very easily here also. So this is a new emerging drug target. Okay, outline of the presentation. So I'll give tribute to a pioneer of PROTAC, introduction, attributes, why protect, mechanism of degradation, tech innovation, and conclusion, challenge, and recommendation. Uh, I was fortunate enough to work with this person, John Houston, in Bristol Myers Squibb. He was the one who started antibody drug conjugate, not antibody drug conjugate, the protein degradation in Bristol Myers Squibb, and then he left and he started first ProTech company in the world, I will say. So he was visionary. He thought like our Dr. Azhar is a visionary. He came from US and Canada and he said, I will set up this thing, okay? He is also like that. He left a very lucrative job. He said, no, I want to do what I want to do, okay? And when we were talking to Mr. Syed, he also is like that. He said he went to very high and dropped to zero and then again started. So he took the risk. Dr. John Houston, he also took the risk. His company is called Arvinas. It is valued at $3 billion right now. And so first compound which he made is already in phase 3 clinical trial in 2019 for prostate cancer. And there is another compound, Arvinas 471. This is also in phase 2 clinical trial. So this is the person who is a visionary. And as I said that I was very fortunate to work with him in Bristol Myers Squibb. And sometime I'm in touch with him. He is the chairman and chief executive officer of a company called Arvinas, okay? So who are these people who thought about this protein degradation in the body? And these are the two academic people. So this is the academic institution. This whole protein degradation came from these two people. You can see their paper. The year it was published in 2001 in Proceeding of National Academy of Sciences. Professor, so I want to give tribute to them because this is very, very new. This is like exploding in US, this technology, which is called protein degradation. Professor Sakamoto and Professor Crew, he thought about it that, okay, every disease is due to proteins. So what we develop a compound or a pill or a drug which will inhibit the protein, production of protein. Why not destroy the protein itself? And you have to be very selective. If you are going to destroy protein, you should not destroy all the proteins which is in your body. So they published the first paper. And so this was a ProTech. In our paper was peptide degrader. Now we are using a small molecule. Peptide have problem with stability and delivery. And second peptide degrader were published in 2008. So now you can see that 2000 and 2001 and 2008, everyone thought, oh, they are useless people. Why they are destroying the protein in the body, okay? And doctors call us to take more protein, and they are calling to destroy the protein. No, but it happens that this became a very, very good way to treat people and do research also. So now, just this morning, Dr. Azhar was talking about post-translational modification. So post-translation modification are, so I will explain more a little bit about that because we just talked very little about that. So I will explain a little bit more that PTM or enzymatic modification of protein after protein biosynthesis. These modification have a lot of things happening there, okay? So I will put the words and you can check later. That includes phosphorylation, acetylation, glycosylation, hydroxylation, methylation, sulfation, ubiquitination, amidation, sumylation, nadylation, isdylation, lipidation, acylation, and methylation, palmitolation, and farnesylation. I cannot explain everything right now, but if you Google, you will find out and ask your biochemistry teacher especially that they can explain all those words. All of them are very, very important in protein degradation. The following event impact the protein of interest the following way. So the proteins are there and these PTM events, what they do. So structure, which is folding and conformation. I talked about alpha fold, beta fold, gamma fold by Google. That is one thing. Function, activity, interaction, and signaling. So these are the protein functions. sequestration, trapping, compartmentalization, and distribution, sorting, secretion, and stability. These are the function of that. So dysregulation of PTM of protein may result in aberrant or disease-causing protein. Aberrant post-transcription factor. Aberrant means the protein is bad. Okay, so this is the word we use scientifically. Okay, so dysregulation of PTM post-translation modification. What they do, they produce aberrant protein because it is not normal now. It is abnormal. So they produce disease-causing protein. So aberrant post-transcription factor could lead to various diseases like cancer, neurodegenerative disease, diabetes, immuno-related disease, and there are many more. So I just these are the diseases we are tackling right now. Most of these proteins are undruggable. So this word I am going to explain a little bit. Only few has been targeted by conventional molecules. So I told there are 23,000 genes. They produce about 4,000 known proteins in the body, and out of 4,000, only 400 are being targeted to treat a disease. What about 3,600? So we call it undruggable because we cannot drug them. We cannot drugging means nothing else but treating them. So that's the word we use for undruggable protein. So therefore, targeting undruggable protein is a new area of therapeutic intervention termed as targeted protein degradation, TDP, or in other words, we call it proteolysis targeting chimera and molecular glues. I will not go into molecular glue. I will just stick to PROTAC. Okay. What are the attributes of targeted protein degradation? So this is already I mentioned. I will not repeat this. Only 400 protein are successfully targeted right now. So why PROTAC? Why not anything else? Because some of the diseases are due to these proteins which are aberrant. So we have to find out another way to treat those diseases. So now we are using this technique. So a small molecule cannot target undruggable protein due to structural complexity and off-target effects. We know that side effects are there. Traditional inhibitor and antagonist only block part of protein function. Occupancy-based PROTAC degrader system. Some enzymes inhibitor have drug resistance. This is happening all the time. You develop a drug, there is a resistance for that. In bacteria, there is a resistance for bacteria. Okay, so we have to develop something that we avoid the resistance, and the drug should survive for longer time. Degraders are active in catalytic manner, reducing the possibility of toxic side effects. So protein degrader, they can recycle themselves. Okay, you give 5 milligram, this is enough maybe for your life or for a week. So that's another advantage of using protein, because protein degrader, they don't destroy easily themselves. They just destroy the protein, which is aberrant, which is bad, right? So they are still there. So this is another advantage. You don't have to take all the time pills. It may be one pill for 1 week. PROTAC can target both druggable and undruggable proteins, and they offer multiple advantages. And there are disadvantages also. The mechanism, how it works. Protein degradation process in the cell. So so it works through a garbage cleaning system in the body. Now, what is garbage cleaning system in the body? So proteins are being generated in the body, but if you keep on accumulating, think about it. You have a kilogram of protein, right? All kind of thing which you eat protein, it has to degrade somehow. So there is a in-body, nature has given us a system, we call it UBQ or UPS system, which is called ubiquitination proteasome system. This is the natural cleaning system of the body. Okay, so this is what a God gifted. Otherwise, we will be having a lot of protein in our body. So that's why we call it the protein can be degraded by 2 pathways. So E3 ligases, these are the enzymes, these are the target protein which we want to degrade. So E3 enzyme is part of this UPS system that binds to the target protein And this is the PROTAC. First, we have to bind this into ligase and then let it go into the body. This will bind this ligase or cleaning system, which will bind to target protein. There will be complex form and it will degrade only the protein, and then your ligase is still, your machinery is still there. So this is, we call it natural garbage disposal system, or UPS system. So again, in this case also same thing happens. It has to internalize, but not through the antigen directly. And there are ways to do that, okay? We have to design the molecule which can directly go inside the cell. So proteasome eliminate the short-lived soluble misfolded protein by UPS system, and lysosome are responsible for long proteins, insoluble proteins, aggregate, and entire organelles. So this is inside the cell it happens. So this field has grown so much that I call it universe. So TAC universe, PROTAC, I call it, okay? There are many things. So there are trivalent protein, bioprotac, RNA protac, aptamer protac, GluTAC, peptac, nanotac, PHETAC, FOTAC, ECTAC, RIPTAC, aboprotac, TRAFTAC, PHOTOTAC, and PC protac. All of them are looking for a bad protein in the body, okay? So this, I said that this field has grown out of proportion, and it's very successful. There are many companies who are working, all big companies are working on this technology. Chemistry-wise, it is very simple. Example of targeted protein degradation, I think it is very faint. You may not be able to see properly, but just tell you that there are these compounds. They are already in clinical trials, okay? And in this case, only the one thing which I will, maybe hopefully I will show, that the UPS system has a binding pocket, okay? And that is a specific, and chemists, they are keep on improving that binding molecule, okay? That binding molecule has to be there. Okay, and there are, these PROTACs are all in clinical trial, and this is very latest data which I wanted to show you. Future of PROTAC discovery, it is not going to be limited to cancer only. It will be going to many other disease areas, and I have already proposed a proposal about a cardiovascular problem. So you can see that earlier it was target protein was these, and there was only one system. Now you have cancer, immunoinflammation. Actually, I'm really interested in infectious disease because somebody was talking about AMR yesterday. That is, and then Mr. Sayed was talking about AMR also. There is not much there. Just only one nephromycin has been approved, and I'm sure, I hope not, that it will get resistance soon on certain bacteria. This has happened to all antibiotics, okay? It is well-known fact. So maybe this technology for AMR, and I will talk to Mr. Sayed about that privately, that we think we should think about that, infectious disease. And we have in Saudi Arabia, we have a huge problem. Dr. Azhar knows that we isolate a bacteria from hospitals, and they cannot be treated with any antibiotics, okay? Even combination of antibiotic. And by WHO, I believe Dr. Azhar can correct me, that after cancer, AMR is the next, that's true. So next most killer disease. We don't think about it, but it is there already. And in neurodegenerative disease and many other more. So I'm looking into cardiovascular disease in this area. Okay. Rest of the modalities, I told you I'm not going to have time. I'm already over time, but I will take 5 more minutes, inshallah. Okay.
All yours.
Okay. No, 1 hour. So I will just wanted to give you the flavor. What else is there in rest of the modality therapeutics? These are the one brief discussion. I'm not going to say anything about that. You can, when you get this slide, please read it. RNA targeted molecule challenges. It's very, very important. These days, RNA is the key now. DNA is old one. RNA is the new kid on the block, I call it. RNA therapeutics, macrocyclic peptides, radiopharmaceutical, and new isotopes are used. Usually it used to be very few, but now there are new ones. Um, Gene therapy and editing, cell therapy, oncolytic, these are highly sophisticated work, which can be done only in very highly sophisticated labs. But I think it is a good idea to think about it now. It can be done here also. That's, I mean, so the sky is the limit, okay? Just artificial intelligence, again, this can be used to improve the drug discovery process, and we are using artificial intelligence in all modalities. So it has a huge effect on us. I will just skip that. You have already seen this before for target validation and other thing. So in summary, I just put this slide that what I talked today is novel chemical modalities. You can see that protein degraders, antibody drug conjugate, gene therapy, protein, oligonucleotide and RNA, and strategic thinking is required in early drug discovery, which one you want to do, and you should know that you can do it. And these modalities are the future of drug discovery and delivery. I will answer any question if you want. It was little heavy, but this is the trend now. So I think you should know and you should learn, and there's a lot of literature already there. Very simple ones are also there. Say it again, please. Yes.
How do we know or analyze that what protein DNA or gene causing the DNA?
So there are tests which are done, okay, and mutations are there, okay, and mutation analysis are done, and in terms of finding how, which DNA, what kind of DNA, where DNA. So you have to do the biopsy of the cancer, and then you will find what is the problem. Yes. Okay.
So we can determine the exact cause for tumor.
It is possible.
Because,
I think there's a huge mechanism is there for that, and you can determine the cause of the cancer. It may be environmental. Now, how you will say, the air is very bad outside, so that may cause cancer also, right? There are many things. Cancer is not one disease. It is a combination of disease. I always say to people when I teach in high school student, it is a combination of disease. This may be your lifestyle. Smoking cigarette, why people get cancer? There is no cancer in smoke, right? So it is a combination of disease.
So one more question.
Yeah.
So if the bacterial resistance develop inside the body, so what can we do to treat the?
You have to start using, I'm not a physician, so don't take my word as a physician. You have to go to doctor and you ask, he will determine whether it is resistant or not. So they will give you some antibody, it will not work, they will give you another antibody, then combination of antibody, okay? And because mechanism for the treatment of bacteria are already determined, gram negative, gram positive, this antibody. Yeah, yes, that's what I said, that you have to go to, and then they will tell you that which, and they will test a number of antibiotic in culture, and one of them will be working, and they will give you. The most important thing is don't overuse antibiotics. I think one question I'm taking from here. Can I take this one first? He was. That's okay. I'm close by. Don't worry.
He's very soft. Sir, one antibody drug conjugate is feasible for only one cancer cell, or will it target entire cancer cells? So
No,
how we will know?
yeah, so I already explained that the cancer cell produces specific antigens, okay, which is not available for other cells. So all other cells then don't express that antigen. Just give you example CD19 or CD8. These are the
Okay.
antigens, okay? So they are on certain cancer cells. I don't remember which are on breast cancer, which are colon cancer. The expert, colon cancer expert is here, so you can ask him. You'll hear in.
Okay.
So these antigens are expressed only on cancer cell, and the antibodies which you are making is going to that, go to only that cancer cell. But it is not easy. It is not just a wet traffic. It has to go through whole circulatory system. So antibody drug conjugate is made in a way that it reaches to target. So it is, you heard about the laser guided missiles,
Yes.
right? These are laser guided missiles. It goes to the right there where it is needed.
Okay.
Okay. Yes. Uh-huh. Yeah.
Uh, so I wanted to ask is what's the because antibodies they have CDR sites, so
Mhm.
like antigen specificity is there. So
Yes.
if we are using mouse-based antibody, how are we targeting it in humans like for drug
No,
delivery?
so at the end, I said that for development of drug, we start with the mouse antibody. But at the last slide, I said that we have to go for humanized antibody. Okay, so we
Yeah.
don't use mouse antibody for human, but in some cases we
Okay.
use
So it's purely for study purposes.
for the development of drug.
Development. Yeah.
Yeah, for the development purpose.
Thank
Sir,
Okay, where?
Sir, I mean, I want to ask, what if PROTAC binds to an off-target protein?
It is possible.
So, sir, how will one recover from it?
So in case of PROTAC, uh, how we develop that the protein is known, okay, and there is a binding moiety, what we call it, thalidomide is the most important. I have given the name there, CRBN, VHN, VHL, okay. So these are the binding thing which binds to the BAT protein, okay. Now, this is, most of the things are initially done in vitro. We find out that, okay, in vitro it binds, what binding moiety we use, okay. So, that VHL or CRBN is important. So, when we put that CRBN or VHL on the drug, okay, and then it goes to the target protein, and we have given TOI means target of interest protein, okay. So sometime it is our body, different kind of enzyme, the drug can be released before. So it is chemist's job to find the right linker, right bond so that it can go to the target protein only. And then there is a delivery way also. It is not like that you take only pill. It can be given directly into the cancer. Most of thing is working on cancer. So there are many ways to it. Is like engineering. It depends if you have a handle on the drug, because chemistry has to work, okay? And we need something on a toxic drug, we need something which can bind to the linker to the antibody. So there are drugs, I know one or two, they don't have any handle. Handle means a nitrogen, hydrogen, oxygen, which should be open and which can be chemically bind to a linker to a antibody through mediamide. Okay.
So are there any such cases when we find that drug is very much effective, potent, but we are not able to conjugate with that antibodies?
It is possible. Means this is also engineering. Buildings fail, so antibody drug conjugate also fail. Okay. One is here. Yes.
We can use a we can use a single drug for the for
antibody drug conjugate, to
Yes,
make
sir.
antibody drug
No,
conjugates.
sir, for treating all the problems, we can use one drug only. So
No,
is it?
no, I didn't say that. I said there is no, I used the word there is no magic drug. There
Yes.
is no magic bullet.
Yes, sir. But my question is it possible, like, can we link antibody with multiple drugs at a
It is,
time?
this is a very, very new, you ask a very good question. Just 3 or 4 weeks ago, I saw a paper. We are putting 2 different drugs on one antibody and sending it to the, but this is treating only one cancer, because one cancer can be treated with 2 drugs. So you have to give, for example, you have to give that drug 2 times. Instead of giving drug 2 times, you put on antibody, both the drug, and then send it. So, and even not only 2 drugs on one antibody, they put 3 drugs, which is called trivalent antibody drug conjugate. This is also happening. It's a very, very, very difficult task, but people are doing that.
Okay, sir. But sir,
It is possible.
yes, sir. But will it not gonna affect, like, one drug will not gonna affect others?
No, it will not, because you are using different. So what happens? Okay, the antibody drug goes inside. Then, as I showed in my figure, it has to cleave. Okay, so one drug goes to the DNA, other goes to the endoplasmic reticulum, other goes to the tubulin. So these are all target for drug. Okay, so they have, but you have to be very smart to do that.
Now you guys are warming up, huh?
I think they took the coffee.
So, sir, my question is,
Mhm.
there is a therapy that is PDT, photodynamic therapy,
Yes.
which are used to treat the cancer, one of the therapy
Mhm.
modality. So my question is, in this method, we are attaching a drug
Uh-huh.
to a antibody.
Mhm.
Is it possible that instead of attaching a drug, we attach that photosensitizing molecule to the antibody, and then we target a tumor, and then we sensitize that molecule to generate ROS and the further degradation of the cancer? Is it possible?
It is possible, and it is happening also. Photodynamic therapy is well known, and people are doing that. Now, your question is whether we can put that therapy on antibody. Yes, we can do that. See, but you have to think about antibody is a very fragile molecule. It is a protein. You have to know the real chemistry to put the, this photodynamic therapy, so that it does not affect the function of antibody. Antibody function is only a carrier, drug delivery port. Okay, so how long change? There are many things you have to do that. In some of the antibody drug molecule, use a very long linker, because the drug was affecting antibody function. So you have to keep away from that, but it is going still. It is bind to antibody. So we have to keep it away from antibody so that it does not affect the antibody function. In some cases, we put a very small linker. That works also. Similarly, not only that, we are putting now radioactive molecule also on the antibody, and that is also working. So there are many, if you look at the clinical trial, there are many things are there. I think I took a really long time.
For the sake of time, we'll move on to the next speaker.
Thank you, sir.
Okay.
Dr. Mohammed
I'm here, you guys can come and talk.
Yes,
okay?
use the lunch time or the other free times to interact. Dr. Mohammed Zainuddin, we have our industry expert. So he has been working in Jubilant Therapeutics, and he has got a vast and deep expertise in the drug development process. So you'll have a perspective from the industry. We have been seeing things, how it happens in the lab, in a university, in academia, but now you'll see what are the possibilities through the industry. And if he allows, you can interrupt during the lecture. Am I right? Yeah. You can be interrupted during the lecture for the questions. So you don't have to hold up even, wherever you find. Okay, while you are dressing up, we'll. Okay, he's ready. Yeah, tomorrow we also have a. So make sure that attendance is. Okay, so yes, so we need to make some